Okay, shall I share screen? Okay. Today's topic is like virtual screening and drug development. In this, we'll cover entire things like types of drugs, virtual screening and docking methods, types, and scoring functions involved in the docking methods, and how a drug has been developed. Okay, let me start. Drug discovery and development. What the term drug discovery and development uh, denotes us. As we think to get a simple pill from, for example, everyone knows Dolo, paracetamol or Dolo or something. Everyone thinks like it's easy to get a pill from a medical shop, but to manufacture that medical to a pill to reach that medical shop, it will take a lot of work and lot many of years and crores of amount for investment. In this, for example, what the I am on the overall steps involved in drug development, it can be seen in the screen. So first, identify a disease, a particular disease, and identify and validate the pharmaceutical target and identify the lead molecules, optimize the lead molecules, and getting the lead molecules for the preclinical trials, and then into clinical trials on humans, both healthy and diseased, and then approval and circulation into the marketing, its formulation and production, and then marketing will be done. So all this takes nearly 15 to 16 years. But the process of identifying and validating the target, identifying the lead molecules, and optimizing the lead molecules has been reduced due to the drug discovery methods like docking methods and simulation methods. Okay, based on these computational methods, application of these computational methods into the biology, biotechnology, and this region, highlighted region has been reduced nearly 2 to 10 years has been reduced in the overall 16 years of uh, drug development method process. You can see the preclinical lab animal testing can be seriously for any one drug molecule has been developed. It has to be, for example, I am defining an XYZ molecule as a new drug. That drug I have identified through some studies or through from literature or from some Ayurveda studies and some references from Ayurveda, some references from Siddha medicine, or some references from the previous chemical compounds. I have defined some XYZ compounds. That XYZ compound should be, if I consider that compound to for a clinical trial, just I have tested on some animal, rat. My studies have been studied. It should be have some positive results, or else it can be considered for the clinical trials. Okay, it can't be considered to study the chemical molecules, chemical moieties to study on human, diseased humans or healthy humans. So, for if I'm randomly selecting any compound, that might increase the chances of getting failure in the preclinical trials and clinical trials. So there is a lot of time waste will be there, a lot of amount will be cost will be wasted. To reduce all this amount and time and manpower wastage, these computational methods have increased the efficiency of chance of getting failures in preclinical trials to lesser numbers. Okay, so this is a step we are supposed to study right now. How we are validating, how we are identifying one disease target, and how we are identifying the lead molecules for that pharmaceutical target, and how it has been optimized such that we can propose that compound can be used to inhibit or activate that pharmaceutical target. Okay, come let's see. So this is what this slide shows the failure stages, both failure and the steps involved in the drug discovery. Okay, and the cost and time involved in the drug discovery process. You can see the x-axis takes more than 12 to 15 years and amount to 150 to 1000 millions dollars. Not in rupees, it is in dollars. So $1,000 million will be we have to spend on a single drug molecule to bring it into a market. See, after defining the drug molecule, preclinical studies and clinical studies in phase 1, phase 2, phase 3. In clinical studies, phase 1, diseased humans are studied. In like, for example, we are studying on some particular type of cancer. Well, the drug we have, molecule we have identified is working on the some but that same cancer. That can study in clinical phase 1 clinical studies. For example, colon cancer. I have discovered some compound, the chemical compound that will act on effectively on colon cancer target. So in that case, in phase 1 clinical trials, colon cancer patients will be taken as a models for study, human models for studying the reactivity and ability of the drug molecule. In phase 2 clinical trials, healthy humans will be considered as a models, human case studies. And then phase 3, they'll be mixed randomly both healthy and human, and then they will study the efficiency of the compound we have defined. And then it will be based on the results from all the three clinical phases, phase 1, phase 2, phase 3 clinical studies, and statistical evaluation based on the studies. It will happen for, see, you can see the duration of the uh, each duration of the each studies here. Nearly more than 6 months, more than 1 year, more than 2 years, and then it will take to the formulation process. After formulation, and it will go for production, and from production, it will be going into market. Okay, so all these involve lot of time, as we seen here, and very lot of amount. So to reduce the failures in these clinical studies, drug discovery process is helping very lot. So to define the drug molecule from the drug discovery method is very important step in a drug development process to reduce the cost and loss of time and loss of money, even the chance of failure. So drug discovery step is more important in any designing methods. Right? Then, so as we discussing, reduce the chance of failures in drug discovery process, loss of failure. So what the failure in drug discovery process or what the failures in drug development? That's nothing but if it is having less efficiency. That means if I am targeting some target protein XY or protein A for colon cancer, if I'm designing a molecule some XYZ, if XYZ is not acting properly or not interacting with protein A as we planned in our studies, that means that molecule is not, that molecule is a failure. So lack of efficacy is one of the issue, and pharmacokinetic properties. The compound what we define should follow the pharmacokinetic properties. So that we will see in later slides. What are those pharmacokinetic properties? And if it's not following pharmacokinetic properties, then it also we can say we can consider this it will lead into the failure in the next steps. And animal toxicity. If the compound what we have discovered is toxic to the animals, definitely it will be toxic to us. That's why animal studies are done in preclinical studies prior to the human studies. And in clinical studies like phase 1, phase 2, phase 3, and before in phase 3, phase 1, phase 2, if it is having adverse effects in humans, male models or female animal like volunteers in clinical studies, if it is showing adverse effects, then also it will lead into the failure of the drug development process. And 5%, as is shown in the commercial reasons, is like marketability. Few drugs cannot be marketable, or few drugs may be specific to some rare diseases or some rare conditions that cannot be marketable as profitable compound like other compounds that are. See, for example, right now, diabetes, insulin is like widely marketed drug molecule. When compared to the other drug molecules, insulin is widely marketed, right? Because more people are affected with diabetes than with some genetic diseases or some other disorders. Disorders will be like, that's for the commercial reasons, like uh, what are the marketable uh, compound that has been prepared or not. Okay, and these are the reasons: lack of efficacy and lack of pharmacologic properties, and if it is having adverse effects in human animal studies and human studies, and a few commercial reasons like marketability and taste, many things will be there. For any syrups, if they are not taste, if they are more order, if it is orderless, it will be fine. If it is more order, some pungent smells like that, then it also will be a reason for abandoning the development process of that particular drug at stage. Okay, these are the possible uh, properties or possible things that can lead into the abandoning the drug development process, or uh, but for that particular drug in the drug development process of particular target. So these will be the major things. Okay, what will be the challenges in the drug discovery process? Okay, first thing is like chemical space and biological space, because what are the compounds we are defining were chemical compounds, but that should be act on the human proteins that defines the biological space. So both what are the compound human proteins that involve the biological space and the drug compounds that involve the chemical space. So both should be valid, and it should be tractable, like it should be traceable, and that both spaces are involved to form a to define a right target for the particular right disease and right molecule for the specific right target. And this defining this right molecule for the right target in a particular disease is the drug discovery process, and this is the best first and best challenge involved in the drug discovery method. Very first thing is choosing a disease. See here we have displayed in this three images. Okay, first thing is like you can see many people like the first image, and with a more obese condition, and second image with a very poor health condition with malnutrition and with more less muscle with like very seriously malnutrition condition, and third image shows the Alzheimer's disease, like shrinkage of the brain and the problem between the connection of the between the neurons. So choosing a disease is up to our interest, and one more thing is like demandable interest. Right now everyone working on the COVID-19 SARS-CoV virus, because the demand is working on the COVID virus to increase the chance of the overcome the COVID virus. So present demand, present choosing disease, everyone is working on SARS-CoV virus. Okay, and next step is like virtual screening. Why virtual screening? Because selectivity is important, because based on the selectivity only we'll get the right molecule. See, in this image, if you are blindfolded, okay, and then opposite person is with a fruit on the head. If he is blindfoldedly passing an arrow, will it be effective in getting that fruit alone without any damage? Very less chances are there, right? Because there might be a chance of getting hurt with the arrow by for that person, opposite person. So selectivity is more important. Selectivity can be done through virtual screening, because only selectivity will define the right molecule. Okay. For example, why virtual screening is important? Why selectivity is important? Because we have one bad experience in the with the history. Example is a thalidomide compound. It's an antiemetic compound. Antiemetic, like it reduces or avoids the sensation of vomiting. Generally, it was prescribed for the pregnant ladies so that they can be their vomiting sensation and nausea can be controlled. Right now, it was banned. Previously, it was used to they used to prescribe this thalidomide compound as an antiemetic for the pregnant ladies. What it led to? Because why selectivity is important and why I'm saying this thalidomide? Because of less uh, selectivity in selecting thalidomide, we had uh, issues with the drug safety compounds, because it led to the defects in the delivered babies, like reduced limb size and absence of the limb. And it was prescribed during early 1960s, and later in late 1950s and 1960s it was banned. It was like they came to know the compound was catastrophic and resulted in this uh, child's birth with limb defects based on the studies. Okay, so at least it was in 40 countries it was sold. Sorry, nearly it was sold in 50 countries as an antiemetic. But after the this studying this condition, they have banned the thalidomide. So selecting a compound is very important. For selecting compound, virtual screening is the best method for selecting the compound. Okay, then what is the virtual screening? Virtual screening refers to a range of in silico techniques. Like it's a range of technique such that large number of compounds from the databases are used to select a very small number of biological testing. Okay, see, for
Excuse me, Dr.
example,
Pradeep.
yes, sir.
Can you please fix your mic? There is a voice error.
Oh,
Yeah.
right now, sir.
While speaking, there is some voice error.
Okay, right now, sir. Is
Your
it?
mic, your mic
Yeah.
is not fixed.
Okay, right now, sir. Is it fine right now?
Still there is a small error is there.
Right.
Noise disturbance backend.
Okay. Hello. One second. Uh, hold for 1 minute.
Are you using headphone?
Sir, is it fine right now?
Yes, it is fine. Fine now.
Okay. Okay. Yeah. What's virtual screening? Virtual screening is a like in silico technique, like computerized technique that involves large number of components from the databases, multiple databases we can select, we can consider, but That consideration is for like to select very few from the databases for the testing between the tar- target. The technically nothing but as biological testing. Okay, this method is like virtual screening. Virtual screening can be done in multiple ways. Like uh, we can define some compounds and then we can synthesize it, and then we can consider for that compound for virtual screening. Usually it will be done in some few, very few pharma labs and some pharmacy departments. They'll be defining new compound synthesizing scheme. They'll write the scheme and they'll synthesize, and they'll using that their own compounds. And then they'll select some compound. We can select some compounds from the existing databases based on the our interest regarding that those are complementary to the targets, or there have been already previous works has been done on the compounds with respect to the same target, or same kind of family targets, proteins belonging to the same family or similar family. So we can consider all those compounds, and they can be considered as in-house database, like our own database, and then we can virtual screen it, or simply we can purchase from the companies, and then we can do the virtual screening with that compounds. Okay, so this in many way we can virtual screen it. And pre-check involves large number of compound information, and particularly to a particular disease or a target, a specific target from the particular disease. Let's see how. Virtual screening. We have two methods: structure based virtual screening and ligand based virtual screening. Okay, what is structure based? What is ligand based? If we are studying the virtual screening with respect to the shape complementarity and existing knowledge of the ligand receptor interaction from the
Sir,
crystallographic.
I have actually, sir, I have one question. Please go to backside.
Yeah,
Yes.
sir. Yes, yes, sir.
Uh, Sir, there is in silico technique.
Ah, yes,
Is
sir.
this one of the method for the prediction or screening?
Yes, sir. Like in
Okay,
silico
so
technique, like the computer in
yes, yes, this
biological
one.
terms, we will define like in vitro, in vivo,
Hmm.
in silico. In silico means what are performing in computerized programming method by using programming or using servers, that will
Okay,
termed as in silico technique. If it
okay,
is
okay.
in vitro, that are
Uh,
done in test tubes or laboratory trays, those things are in vitro techniques. In vivo techniques like directly on the animal cells.
so for this technique,
We need
there are
system.
several softwares. Can
Yes,
we
sir.
use anyone like R is there, Python is there,
Yes, sir.
MATLAB?
That's what next slides were.
Okay, 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 sir. Thank you.
Yes. Okay. So anyone can raise at any point. That will be easy to clear, and then we're going to move forward. Yes, sir. In known structure-based virtual screening method, we have the knowledge of the receptor structure and receptor geometry, a receptor of the target's structural information. Known active sites will be there, or binding sites will be there, or we can get the all possible residues that are involved in ligand interaction with the crystallographic structure interactions, or that has been co-crystallized with the receptor. Okay, so all this information is known. So with respect to the structure, we will search the compound. In this case, is a structure-based virtual screening method. And in another case, we'll have no knowledge of the receptor, and we'll have the only ligand. What we have or what we have, we'll design or we'll define based on the requirement of the interactions mentioned in the theory, theoretically mentioned between the interactions. Okay, in that case, if we have the knowledge of only ligand, we'll define the mathematical model, nothing but pharmacophore model. Okay, in that again we have energy-based pharmacophore model and a normal pharmacophore model. Okay, from those pharmacophore models, we'll search for the new compounds or from the existing compounds from the databases, and then we'll define respective to the target. Or else, if the properties are also known, IC50 values are also known of the ligands that we have, then we'll go for the QSAR studies, quantitative structural relationship studies between the those compounds and the receptors, and that also will have a 2D QSR and 3D QSR. So based on the dimensionality also, quantitative structural relationships can be studied. So these methods is the ligand based methods. Okay, simply the structure based method we'll say as protein ligand docking studies. Fine. And then if it is ligand based methods, we'll have the actives are known. If actives are unknown, not and inactives are known. In that case, similarity searching will be done based on the ligand. That ligand we have will be used search for similarity compounds in the databases we're considering. And in case we also, if actives are known, we can go for pharmacophore mapping, as I said. And if actives and inactives are known, both are known, in that case, as Sar was asking, we'll go for machine learning methods. Okay, in this we can use Python, and we can go for machine learning techniques are there that will be seen in the later slides. Okay, it'll go with the similarity searching method. Okay, rationale for the similarity search. In this, you can see the three compounds I have displayed here. One is morphine, one is codeine, one is heroin. So both are biologically active compounds every time, and they are more chemically active compounds also. Okay, well, the principal state of these molecule structures will be similar, right? But its medicinal values and medicinal properties are different. Morphine will keep us to sleep. Heroin will keep us in hangover, right? Say drug, actually all are drug, but these heroines are the banned compounds, illegal compounds. Like, legally they are banned. Okay, fine. But the basic structure are same, but the medicinal properties are different. You can see the difference here with the functional groups and moieties group difference. Let me show it. One, two, three. Can you notice the difference? So these functional group differences, this moiety difference in the difference to the main principal or backbone structures will change the compound properties. And those are medicinally they lack more active or they are completely differ with the sorry completely differ with the main principal compound. Okay, so similar searching method, if we take one compound and we'll have to search for the similar compounds, then we'll get Main backbone structures will be same with extra side chains. So this searching will differ in, like we can both based on geometry based you can search, like it will whatever we are considering for searching in that compound, geometry will be searched. It will not define on look out on any functional groups or any functional matrix. It simply look of the geometry of that compound. If it is a polygonal in geometry based search, if it is like this, sorry, okay. For example, let us say this is one small compound that will not be exist. Let us for say think for example in geometry based search, similarity search, this this will be the geometry. Okay, so this shape will be used to search. It will not look on any functional groups. So only this shape, what are the compounds matching to this shape will be considered in geometry based similarity search. Okay, but in case of if we are considering only geometry, then we will not get any important compounds, right compounds. So for measuring and defining the compounds, we have some descriptors. Okay, like three components of a similarity search. One is like physical properties, second one is 2D properties like fingerprints and indexes and substructures, that is common substructures. And another the third one is like 3D properties like 3D fingerprints and molecular fields. Okay, so based on physiochemical properties, we can search. Similarity search can be done other than geometry or shape-based similarity screening. As I said in previous slide, geometry-based screening will just or shape-based similarity screening will just look on the geometry or the shape that are complementary to each other, but that will not look on the functional groups here. As in this these functional groups, see the change in the small functional group moiety will change the compound, right? So we'll not get the similar, we'll not get the same, but we can get the similar. So for searching the similar compounds, we should concentrate some other properties that are molecular descriptors, like physiochemical properties, that in like example molecular weight, log p value and pSa values, and 2D fingerprints and topological indexes, and 3D fingerprinting and molecular fields. And we have one more quantitative measure value, quantitative measure of the similarity, that is nothing but similarity coefficient. And this similarity coefficient between the molecular descriptors we consider is also plays a major role in defining the similar compounds. Okay, and what is 2D fingerprint? In 2D fingerprint molecules, it represents the binary vectors. Okay, binary is nothing but zeros and ones. Okay, like bit strings. Uh, bit scores nothing but zeros and ones. Bit strings is nothing but a series of zeros and ones. For example, if a zero is mentioned as an empty white and one is mentioned is one, you can see the bar white, black, white, black, white, 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 black, like this 0, 1, 0, 1, 0, 0, 0, 1. What this 0, 0, 1 represents? The bit scores of the string molecule represents the presence of the fragment that we are considering in the compound for as a template for searching. And that zero or white fragment represents string represents the absence of the molecule, like fragment of that molecule. You can see this carbon, alpha carbon is bonded with the carbons in all four sides. That carbon is represented by the presence of one that has been divided the string in black. You can see this simple concept in barcodes of your any product. If you any buy any simply uh, food, biscuits or chocolates packet, you can see this barcode graph in the backside of your packet. You can see these lines bar. So this represents the quality and the FSCA numbers and the things of that number defining the zeros and ones information regarding the product. Here we are defining these zeros and ones for the presence of the fragment of that compound molecule. And like this, each string will be defined for the entire molecule. And that string is used to search. We are not considering the shape. We are using the zeros and ones strings. These are zeros and ones are bit scores. So these bits as a string we are using for the searching. So on these big strings are used as a binary vectors for searching the databases. Fine. Based on this, we can define the two structure commonality between the two structures. And then, for example, we can see the fragments. How we can define this fragments. Okay, let's move this. Yeah. Here in these fragments, you can see these augmented atoms and atom sequences and bond sequence and ring composition here and ring fusion here and atom pair and these are mentioned as a fragments. So in these uh, fingerprints we have predefined fragments each with will be the single bits and this here we are representing here are the MAC keys. Okay, so we are defining the fragments into fingerprints here. Okay, here we are hashing the fingerprints. In this, we are combining the fingerprint. Here we are combining the both fragment method and okay, क्या हो गया? Let's wait. It is some.
It is taking some time in the opening.
Yeah, yeah. Okay, someone asked about some light on 2D fingerprinting in practical. Thank you. Yes,
Yeah.
okay. Okay. But right now we have some I don't know, I forgot his name. Okay, let it be.
Uh,
Okay. The for the person who asked that about the fingerprinting, I'll explain somewhat more. Okay. Good to hear from you, Mr. Sorry, Mr. Mrs. I forgot. Good to hear from you, participant. 2D fingerprint is a basic method we use for similarity searching. Okay, but we have more advanced method right now. Then uh, previously we used to search based on the fingerprint methods because we have less number of software availability and less number of programming availability in like application into the bioinformatics. So we use a more fragmentation method, and then after fragmenting the compound, we'll be defining into fingerprint method, fingerprinting, and then that fingerprints were applied to screen the compounds. Okay, but right now we have a more application oriented programming and more advanced softwares. We use pharmacophore screening, QSAR screening. Okay, so in pharmacophore we specifically define features like acceptors or donors, those things, hydrophobes and nucleophile things, and then we will be searching based on that. So in that case, rather than going for 2D fingerprinting, we have more advanced searches. Okay, so
Sir, please
yeah, you
remove
can.
this screen. We can see only white screen.
Actually, it was hanging, sir. Okay, I'll just close the program.
Yes,
Okay.
close and then reopen.
Yes, yes,
It's
that will.
fine.
Yeah, I don't know why it's happening. Okay, and this is what we are mixing the hashing method and then binary code method. So in this, both we will consider the non-linear compounds also will be considering in this. So in this we'll get like daylight method and unity fingerprint method, and I'll explain how and what are those. Okay, I didn't mention here, so I'll explain from this image itself. Okay, unity fingerprint methods are by like it will define the both presence of and absence of the fragment molecules as explained before in this slide. Mm. In this 2D fingerprint method, so we are just uh, along with this 2D fingerprint method, we are adding the knowledge of this fragment method in hashing fingerprint method or unity fingerprint method. Okay, based on this bit score, along we'll have the hashing method. You can see here star. This defines the this is important moiety of this molecule. It has been fragmented. Okay, based on like both in this type of screening, both this by binary string bit score string along with the unity score like hashing, both should be matched. Then we will get the similar compounds. Okay, Dr. Grace. Sir, which one you asked me to repeat it, sir? Sir, Dr. Grace uh, asked me to repeat about which slide, this slide or previous slide?
Dr. Grace, can you please repeat your question?
Dr. Grace.
Yeah, Dr. Grace. Also, you can unmute yourself
Yeah, you can
for your
ask.
question. Yes, you can unmute.
Okay, thank you very much. No, I wanted him to repeat where we lost when he was the screen was white. So I wanted him to repeat. I have my questions, but I will say that at the end of the lecture. Hello?
Dr. Pradeep. Pradeep, you got it?
He can go on. I will watch the repeat, the video later to just,
Okay, okay.
yes.
Okay,
My
thank you, Dr.
questions,
Reyes.
yeah, my questions will come at the end of the lecture. I have them written down.
Okay.
Okay, I thought you can, we can clarify the question right now so that not be doubt will not be continued further for this.
Okay, if you want me to ask, then
Yeah.
I followed up the molecular docking studies, although we may not have reached there, but I'm just wondering,
Yeah. Okay.
so the disease must be traced or linked to protein. It must be protein in nature for you
Okay.
to use a
Okay.
molecular docking tool. What if the disease is not protonic in nature or protein in nature? Sorry. What if it's not protein in nature? So how will you be able to, I mean, select the receptor, the molecule to, dock with the ligand? That's my question. I hoped
Okay.
to, yeah.
So let's say what I understood your question is like we are having some target protein molecule in natural environment. We can't imitate that natural environment, like, right?
Yes,
How?
what if the
Uh,
disease is not protein in nature? Cancer molecules, yes, you can get some of them from the PDB, but what if it's something like malaria or something that is not, I don't know if you understand what I mean.
okay. If you the protein, some disease causing, we have some disease that
Yes,
caused by some protein. That protein has no structure,
exactly.
right? Actually, I think you might be missed yesterday's session. That session, yesterday's session was meant particularly for this doubt only, particularly for your doubt. In that session, we have explained how to generate a model, a PDB structure,
Okay,
three-dimensional structure for the proteins that has no structures in the PDB.
thank you, thank you.
We can build, if we need, we have only amino acid sequence or protein sequence for any particular protein, we can generate three-dimensional model. That's what the yesterday's session. Is that clear, man?
Yes, thank you very much.
Yeah,
Thank you.
welcome, ma'am. Okay, we'll go with this fingerprinting method. And this in this fingerprinting is one of the method for similarity search. Okay, we are including both hashing with particular properties and then with the fragment presence of the fragment particles fragments in the molecule in the form of bit strings, zeros and ones. Okay, and that fingerprints are used to search the similarity with the compounds. Okay, and another method is like substructures. Okay, substructures are nothing but this is one structure. In this structure, we'll define particularly important, or they are necessary for the interaction with the compounds based on the reactivity of our knowledge or the chemical knowledge or molecular interaction knowledge of this particular ligand. Okay, in this we'll have adopt a Morgan algorithm such that we'll define the compounds, substructures, descriptors particularly. In previous methods, we will define the descriptors for overall compound, but here we will define the particular substructures descriptors than the entire compound descriptors. And these substructures will be defined, and those substructures will be used to search for the compounds from the databases rather than the entire compounds. Okay, this is circular substructures. And then I defined base apart from the uh, properties, molecular descriptors, similarity coefficient is important. Here in similarity coefficient, we can define the similarity coefficient or Tanimoto coefficient, nothing but similarity between the properties, molecular descriptors between the template compound what we used for searching and the compound hit compound received from the database. You can see one of the upside one. This compound will be like template compound. This as a template compound and this as a hit compound resulted from the search. We can compare the bit scores here, zeros and ones, bit strings, and this bit strings and the property or similarity. How we calculate the similarity coefficient? You can see here. From this 100% matching will be the same. Same compound will be considered here. But we are searching for the similar compounds. Based on the Tanimoto coefficient or nothing but similarity coefficient, we'll search compounds with less. So you can see the coefficient with less percentage. We'll get the compound like this. If with the compound with we are searching for the uh, 20% confidence with the Tanimoto coefficient, similarity coefficient, we will get the compound like this. You can see only some part will be matching, only some backbone portion or some side chain will be matching. If increasing the percentage of similarity identity, you will getting the compound similar to this. You can see the structure here. 80% similarity coefficient is there. We are getting the compounds, backbone compound more or less almost similar to the compound we consider for the search. Okay, if you are considering this compound as a template or existing inhibitor or existing drug molecule for searching into database, we consider with more Tanimoto coefficient similarity or similarity coefficient will like this will get similar compound as displayed here. And scaffold hopping is one of the method, good very good method to identify the new compounds with this like defining the structurally novel compounds by identifying the central core of the structure molecule. Okay, that also will be explaining the next slides. You can see the one of the Smilog keys are there. In this Smilog keys will be defined the vector pharmacophore vectors in the form of keys. Atom typing will be done for the properties like hydrogen bond donor and hydrogen bond acceptor and bulkiness or heaviness on electropositivity. And all these will be given the DAP keys. DAP keys is nothing but strings, bit strings. Bit strings nothing but bit scores with help of like combined bit strings with that defining the vectors. Okay, and that will be termed as Smilogs. So this is the Smilog key for this compound displayed here. And reduced graph method, both are here. Uh, this also defined based on the Smilog keys method. We can define the reduced graph method. This also these are previously used methods for screening the compounds. And 3D fingerprinting method. As previously we discussed, 2D fingerprinting method. Here only two-dimensional structure is considered for defining the presence and absence of the moieties of the fragment moieties in the molecule. But in case of 3D fingerprinting, we will be studying the presence in both the considering both XY dimension and Z dimension also. presence of that molecule in that position. So that distance range, the range and the angle also considered in the for the donors or acceptors, aromatic centers, and the hydrophobes or hydrophilic centers of the ligand molecule. In this, torsion angles plays an important role in 3D fingerprinting, and this 3D fingerprint is considered for searching the database. Okay, and alignment based 3D similarity based. In this, as I very first explained, shape based similarity search, nothing but geometry based similarity search. Okay, in this, ROCs will be there, nothing but rapid overlay of chemical structures. In this, only common value will be there like volume that is more very close to the existing compound we consider. For example, we are considering this compound for geometry search, shape based search, then the confidence level is near and more which compound search has more confidence to the volume that is similar to the template structure will be considered as a best one. Okay, yeah, because in this concept, same structure will have the same function. So on that concept, this method will be executed. Okay, in this we have to consider the torsion angle flexibility for considering the presence of rotatable bonds between the molecule atoms of the molecule. For example, we can see the torsion angle here in this might be rotated to this, right? If the distance between A and D is reduced, the B and C is also reduced, then the shape or and the position of the atoms will be modified, right? So while considering the shape complementarity or while defining the molecule for considering the screening process, we have to consider the rotatable bonds also, because when rotatable bonds are present between the atoms, if we are defining into 3D descriptors or 3D fingerprints, the rotatable bonds may change the optimal distance or may not change the optimal distance while screening process. But in case of virtual screening docking method and in dynamics process, this will may change and it will induce some adverse effects also. So while defining these things, we have to consider this such that combinatorial search will be executed. So confirmation of the compounds also important in defining the fingerprints method. Okay, and then we have seen like similarity search method. Similarity search method we have seen like 2D fingerprinting, 3D fingerprinting, and smile keys, and lastly flexibility, confirmation flexibility, and shape-based similarity search. But in case of pharmacophore mapping, we'll be defining the pharmacophore nothing but an mathematical model that representing the features features of the intramolecular features of the compound like hydrogen bond donor or acceptor and hydrophobic feature, aromatic ring or the projected point here, and hydrophobic point, nucleophile, neutrophiles things. Those things will be defining, and the angle between the one functional group, one functional moiety and another functional moiety feature will be defined. The angle and distance between the pharmacophore feature like hydrogen bond donor, hydrogen bond acceptor, or hydrophobe, or nucleophile, or aromatic ring. All these features, the distance between the features and angle between the features also plays a major role in the pharmacophore. So pharmacophore is a representation of internal features and its representation mathematical model is nothing but pharmacophore. Pharmacophore we have many methods like E pharmacophore and energy based pharmacophore and pharmacophore, ligand based pharmacophore. Okay, here trying to predict the ligands how it will bind to the receptor without knowing the structure of the receptor. Okay, in the pharmacophore model we are exploring the pharmacophoric features that can be possible in binding the or involved that can be possible in interaction with the receptor, and those features will be represented in the pharmacophore models. So thus, without the knowledge of the receptor, we are defining the ligand-based pharmacophore and structure-based pharmacophore. So nothing but energy-based pharmacophore method. In this, the known ligand and known receptor were docked, and interaction energy between the structure and receptor, that the receptor and the ligand is considered, and those features that are involved in interaction will define the pharmacophore features of the ligand. Okay, in this known interaction will be considered. So one example here in pharmacophore searching, if we define one mathematical model, one pharmacophore model with some features like hydrogen bond acceptor and donor aromatic ring, hydrophobe and hydrophilic nucleophiles, with that feature, for example, it has only four or three pharmacophoric features, and that three pharmacophoric features is searching from one pharmacophore considered for searching against the database. It will search against the pharmacophoric features, not the structures based on its presence of fragments or presence of moieties or its shape complementarity. Nothing it will check. It will check for the pharmacophoric features. Hydrophobe is present. Hydrophobe is present here or not. Nucleophile is present. Here nucleophile is present or not. Here acceptor is present. Here acceptor is present or not. And then it will check with the distance between the pharmacophoric features. Is this similar to the compound we have pharmacophore we have considered for searching or not? And if it is more or less similar, and it will consider for the angle between the pharmacophoric features, and those pharmacophoric features angles, distance, and torsion angles, everything will be considered, and then the compound will be retrieved from the database. So when compared to the older methods like 2D fingerprints things, pharmacophoric based search will increase the pharmacophore based method will increase the ability to retrieve the specific compounds, more specific compounds from the database. This is one of the example. You can see the features of A, B, C, and the distance like some angle, oxygen, and nitrogen. From compounds, different compounds having a different angles and distance. Okay, based on like the features, distance between the features and number of features and the torsion angles between the features will be considered for retrieving the compounds. Okay, and structure activity relationship. This is if we have a known structure and its properties are known, then we can go it for the knowledge of known will be implied to the knowledge to predict the knowledge of unknown. Okay, nothing but structure active relationship. In that we have QSAR, quantitative structure activity relationships, and it was uh, restricted to small compounds, but to homogeneous compound it can be optimized easily, but it has been developed into 2D QSAR and 3D QSAR techniques. 3D QSR is widely used and it is more preferable than the other techniques because three-dimensionally we are using the structure and its known activity, IC50 values, are compared with the expected resistivity values of the known compounds and the known inactive compounds, and that will build some model with the respective to the activity and the structure, and that relationship is modeled as a QSR model, and that QSR model is used to search against the database such that QSR models, similar QSR compound having the features representing the QSR model will be retrieved. Like this, we can retrieve the uh, compounds from the database based on the QSR. And machine learning method, one other method is machine learning method. Generalize, I'm explaining the very outline of the machine learning methods. I'm not going deep into this. Uh, okay. In this, for example, we have substructure analysis. We can perform recursive partitioning, and we can perform SVM method like support vector machines. We can get the k nearest neighbors, and we can go for the neural networks for the defining the model activity on the defining the compound properties and defining the new structures from the existing structures, to define the pharmacokinetic properties from the existing compounds, to define the to make a model from the unknown based on the known. We can define the properties for them, predicted activity values we can define, and then we can consider, we can reduce the compounds from large databases to very small, such that we can reduce the chance of getting failure in the drug development in the clinical trials. And all this can be done through neural networks and SVM methods. Okay. And it's very like, we are not going deep into the machine learning methods. We have actually, this is very basic methods I have mentioned here. We have many methods are there. Okay, like substructure analysis methods. Here, unknown molecule is scored with the weights. They'll be giving some weight and score to the molecules with the known active, known each fragments, and those weights are those scores were considered to get the training set and test set. Work on the test set based on the training set based on the test set, and those weights were summed and against the test molecules will be probably define the test set, and then reduce, they'll be reducing the they'll be considering the probability of the activity. Okay, and the calculation of the weights, nothing but the scores. They'll be given the typical example is like ACT1 active and active plus inactive. Okay, like the number of molecules are active and number of molecules are inactive, both. So this will give the uh, weight, nothing but score of the each fragment substructure of the entire molecule. Like this, it will be calculated for the entire molecule, and then recursive partitioning. In this recursive partitioning, we will define a decision-making method, like to generate a good model with more and poor predictive power. Like it will be split into the more uh, trees, and it will be statistical method. It will split into multiple parts, and then again it will be rearranged to get the like iterative method. Like repeatedly it will split, and while splitting it will making a decision, it will remove the unwanted things that are not easy to interpret or not easy to take a decision or easy to validate. So like uh, removing the compounds that can be not validated, not can be kept into some decision that can be derived to decide. So like this, it will be kept on removing the things, like it will make a like decision tree, it will develop, and like partitioning, and then collecting it together. That's the recursive partitioning method. Simply it will split, and again it will combine and split and combine, and this method will be repeated until it gets a very few. that are similar to the like required compounds. For example, we have 50 actives and 12 inactives. In this, based on the properties of polar surface area, it is we have less than only 5 here with dipole moment and uh, 9 inactives with the dipole moment less than 5. In again, we have 0 with the dipole moment inactives is less than 5 is only 0. And in case of inactives, higher than 5 is 9. Similarly, you can see the uh, other property like principal axis length. Compounds with the principal axis length of actives were 45, inactives were 3, and C log P. The compounds with C log P mean like principal axis length less than 7 is 3. And again from 45 with the compounds with having C log P is less than 3 is 0. And greater than 3 is having 5. Like this, it will split based on some other property. Again, it will combine and it will split to decide the decision making tree such that more compounds can be dropped out from the tree and less number of compounds can be retrieved. And structure based method, a known structure based virtual screening method is like simply protein ligand docking. And what is protein ligand docking? How does a ligand bind to the active site of a protein? And what's the docking algorithm does? And what is the scoring function uh, says about the docking? Docking algorithm is based on the two key components. One is algorithm used to search and scoring function used to define the docking interaction. Okay, in searching algorithm, that is nothing but algorithm that generates the poses, nothing but confirmations or positions or orientation of the ligand with respect to the receptor active site or receptor geometry, that is both are complementary and both exist as a stable complex, not only just a shape complementarity to the active site of the or the deep valley active site valley residue deep curve of the residues in the protein. It should be have some interaction and it should have possible and stable conformation, and also it should be shape complementarity should be there. Okay, that searching method is nothing, that searching algorithm is like important thing in any docking method. And another important thing is like scoring function. What is the scoring function used to define the docking method? That defines the that also plays a major role in defining the docking method. To identify the most likely pose of a molecule that is complementarily to the receptor, and to define the best ligand from the all diverse compounds we are considering for the docking process, nothing but the binding affinity can be defined with the scoring function. Okay, for any docking method, searching algorithm is important and scoring function is important. And examples of dock searching algorithm is nothing but dock. The first docking program by Kunz et al. In 1982. This was completely based on the shape complementarity of the receptor and the ligand. In this, both the receptor and ligand were rigid. That means there is no movement and no flexibility will be considered in this algorithm. Only based on the shape it has been, it will dock. And current we have many multiple algorithms like fragment based algorithms, Monte Carlo simulated annealing algorithms, and genetic algorithm and systematic search algorithm. Fragment-based method algorithm is used in some docking softwares like Flex and dock upgrade version, and Monte Carlo simulated annealing method algorithm is used in wide known docking software Autodock, and affinity based ligand fit dockings used in auxiliary softwares, and genetic algorithm is used in gold and older versions of the Autodock, and systematic search versions algorithms are used in OpenAI softwares and Glide. Glide is another algorithm used in Schrodinger, very commercial software used for very well-known docking software. In case of dock, developed by Kearns et al. 1982, this algorithm is completely defined on the shape complementarity. In this, both the receptor and ligand is considered as the rigid compounds, and now flexibility is considered. But in case of the cavity filling, we can see as examples like spheres filling it into the negative image as shown here. You can see the spheres of different sizes varying with the respective to the fragments of the molecules in the compound ligand compound, and that spheres are fitting into the geometry of the receptor as shown here. You can see that each touches the surfaces at least 2 points. That means this compound, the ligand compound used in this should have at least 2 points of interaction with the receptor. That means that interaction, that centers of those pairs will become the potential location for the ligand atoms to bind. And this is the algorithm they used in dock. You can see ligand atoms that are matched to the spheres. Okay, we have seen that complementary to the receptor, and those atoms that are equal distance between the spheres that are used in the those compounds were matched to the ligand active site, and if there is no steric clashes, and that ligand is scored as a best ligand, and the differences are mapping or termed as the poses of different possible poses, and each pose is scored based on the fitness between the receptor and the ligand, and high scoring pose or highest scoring best fit ligand is represented with the user, such that it can be used as a that can be considered for the further studies. But in case of exploring the conformational space in dock, we are using only geometry space, not we are not considering any conformational spaces or rotational bond spaces. If we have to consider those things in docking interactions, then we have to consider the conformations of the each molecule, each atoms, and then ensemble of that conformation of the atoms of a molecule should be considered, because then only a series of conformations can be generated before docking to the receptor, and each conformer is docked in such a rigid body, and then it will be optimized or approximated or averaged to the conformations of the interest, and this is done in many glide also. Glide I mentioned in the as in Schrodinger. If conformation space explored at runtime, then the accessibility of the conformations is explored more as the time at the same time of the docking. and that is done in genetic algorithm, and that is used in the gold software. And similar thing has been done in based on Monte Carlo simulate annealing algorithm method, and that is used in the Autodoc software. And similarly, incremental construction method has been applied in the FlexX docking method. You can see the genetic algorithm method here. Complete population of the database will be considered, and that has been evaluated based on the interaction. That is a confirmation that are binding, and that confirmation binding with the receptor will be defined. Okay, and then that pose is again reselected, reoriented with the ligand confirmation with the binding site of the receptor again selected, and then again it was mutated, like again it was repeated. One ligand pose will be mapped, and then again it will be repeated by changing the van der Waals internal energy, or by changing the functions used in that scoring function used to define that pose, and then again it will be repeated. Similarly, these iterations will be done until an optimal scoring function is obtained, and that optimal score that has been received will be defined as the best pose. Someone has some doubt? Yeah, I think Anaya Chinesa, these are the docking algorithms I'm explaining here. You can follow, I hope. And next to genetic algorithm, we can see the example here, how the docking receptor example, how the backend is done. Okay, receptor ligand compounds the difference with the molecules atoms, and how it is modified here. You can see the backend programming here, and this summation of the van der Waals distance. Ij defines the position of the atom. Okay, receptor and ligands, atom X at the position of I and J, and this van der Waals interaction, van der Waals diffraction, attraction forces. All this we can see here summation of empirical formulation. Okay, so this is how it will define the process step by step, and it will be repeated and it will be iterated again to get the optimal score. Okay, see, four ligand atoms at the same time it is calculating in this, right? Template, template X, Y, Z has been it been calculated at the same time. Again, one with the again with the remaining things. You can see the distance changing like this. It will be summated, and summation of all these scores will give the scoring function of that. So for better understanding, you can see here. This image will show the receptor and ligand, receptor in red bubbles and ligand in the blue bubbles. You can see here receptor atoms and ligand atoms. See, first atom will compared with the ligand atoms, possible ligand atoms, all the ligand atoms, and second, third, it will consider one round will be the one iteration will be done, and second atom, another iteration will be done for all the ligand atoms. Third atom will be iterated with all the ligand atoms. Repeatedly, it will be done for all the ligand atoms with the all receptor atom with for the possible for here the possible all ligand receptor atoms will be repeated with the all the possible ligand atoms such that shape complementarity is attained and stable conformation is stable conformation has been arrived by the ligand with respect to the receptor. Okay. And in case of flexible docking, we can consider, you can see this. Okay. In case of flexible docking, you can see this. In this, all the torsion angles, rotatable bonds, and the possible rotation bonds will be considered as flexible. They are allowed to rotate into uh, its possible distance that can rotatable without changing the backbone of the ligand, and that flexibility is considered as a important thing in defining the interaction, because the flexibility will increase the ability to interact with the receptor. So flexible docking is more considered better than the rigid receptor docking. In case of energetics of the protein ligand binding, I can see this scoring function here. Total binding free energy, because we should have considered the bonding interactions, hydrophobic interactions between the ligand atoms and between the ligand and the receptor atoms, and van der Waals interactions uh, present between the ligand and the receptor, and conformational changes that generated the energy between, changes the energy between the potential energy between the potential energy of the complex, and loss of entropy due to the involvement of ligand binding to the receptor. And this can be given simply summation like delta G bind as equal to, will be defined based on the delta G of the solvent, delta G of the conformation, delta G of the interaction of the ligand bonding, and delta G of the binding free energy of the rotational angles, binding energy of the torsional angles, and binding energy of the vibrational angles. All this is considered as to give, we can define as the binding free energy as delta G bind. So docking energy, along with the docking score, we should define the binding free energy. Ligands with more binding free energy are said to have high docking ability, binding affinity towards the receptor. So delta G bind along, delta G score along with the docking score should be considered. And scoring functions, as I said earlier, for docking studies, docking algorithms and scoring functions are more important. As we studied the docking algorithms uh, previously, we'll see the scoring functions here. In this, we have molecular mechanics force field based scoring function, and empirical based scoring function, and knowledge based scoring function. In case of molecular mechanics force field based scoring function, the score will be estimated or calculated based on the summation of the intramolecular attractions along with the van der Waals and electrostatic interactions for all the atoms between the ligand and the receptor as a complex. Thus, molecular mechanics force field scoring function is said to be a good scoring function, and this scoring function is used in charm and amber force fields. Charm and amber force fields are used in Gromax molecular dynamic simulations method. Charm is commercial, amber is also commercial force field, not freely available force field. In case of empirical based method, it is like summation of all the interactions like both hydrogen bonding interaction, hydrophobic interaction, van der Waals interactions between the ligand and the receptor are considered. That means empirical summation is done. Summation of all the energies is considered such that to give the score, empirical score of for the binding interaction between the ligand and the receptor. And this is given as a glide score and chem score and orthodox score. Orthodox score is given in orthodox, and glide score or XPG score is given in Schrodinger, and chem score is given. And in case of knowledge based method, it is a completely intramolecular interactions only is considered, and that define the potential energies between the intramolecular interaction only. It will not consider the other non-bonded interactions. And this is the statistical method, and it is given in as a drug score and PMF score. Among these three scoring functions, empirical scoring function is considered as the better one and the best one because it considers all the interaction methods to define the energy of the interaction, the complex. In case of empirical potential energy function, as I said, is the best scoring function than the other scoring functions. Here you can see this. What are the energies it is considered? So bonding interaction it is considering. You can consider the green balls as the two atoms of a molecule, and the bond distance between the two atoms is considered. Uh, Bond angles between the atoms is considered, and dihedral angles and torsion angles between the atoms rotatable bonds is considered for scoring function. And also the electrostatic attraction and van der Waals interactions, force of attraction between the non-bonded atoms is considered to define the scoring scoring of the interaction. Thus, empirical potential energy scoring function is considered as the better and the best scoring function than the other one, other scoring functions. You can consider these bond distance and bond things like for the bond vibrations and the angles and the header angles and torsion angles for the bending vibrations and rotation vibrations, energies and electrostatic and van der Waals interactions for non-bonded interactions. And this also gives the entropy and enthalpy of the considers the entropy and enthalpy of the interactions. And ADME properties of a drug molecule. Ideal properties of a drugs. ADMET is nothing but absorption, distribution, metabolism, oxidation, toxicity. Okay, let's just recap the previous things. Like we have seen the compounds, I mean target we can define from the particular disease. For that target, we need some compounds to dock. We can consider compounds of known, unknown, the known values based on pharmacophore search and 2D fingerprint search, or with based on the structure knowledge, we can define the go for ligand screening. After virtual screening, we'll get the compounds, and those compounds can be docked to the receptor based on the different algorithms, dock algorithm and flex dock algorithm and genetic algorithms, and we can use dock to the receptor. After docking, we can get the analysis based on the scoring function. Knowledge based scoring function, Monte Carlo based function, knowledge based scoring function, empirical scoring functions are there. Among the empirical scoring functions, each and everything are considered. After defining the best compound with the best docking pose, best or favorable docking conformation with the highest docking score and highest, best binding free energy, we should check with the ADMAT properties of the compound such that it can be used as a drug molecule or not. In that case, ADMAT is defined as absorption, distribution, metabolism, excretion, and toxicity. Okay, absorption defines the ability of compound that can be absorbed or that can be carried into the bloodstream such that to reach the region of site of action. In case of distribution, gets to the target tissue. That is, after crossing, it should allow the blood-brain barrier to reach the brain. In case of brain, is a site of action, and distribution involves the distribution of all the drug molecule to the all the regions of the parts site of action. And metabolism, if metabolism is not readily metabolized, it should be excreted. And metabolism defines the how much it will be metabolized at the site of action. And excretion is not, if it is not metabolized or not showing its activity, it should be excreted. I mean, it should be secreted from the body. Okay, and that defines excretion, ability of excretion. And toxicity, if it is non-toxic to the tissues or not, should be defined. Then only it will be considered a drug molecule. If it is toxic with means it's not toxic with lesser side effects, that also considered based on the beneficial properties of the compounds or beneficial properties of the drug molecule in the clinical trials. And these are over administration of the drug. For example, oral administration frequently brings the drugs to the portal system to the liver. From the liver, it should be excreted. Okay, and this the ideal properties of the drug. And these are the drug factors affecting the drug response. If we are defining with the best force, best compound with the best force and best binding energy, and having the best ADME properties, but it is having less absorption and less distribution, or not able to properly eliminate it from the body, or having a less pharmacological effect or less clinical response. In that case, if it is more toxic, then it is said to be the we can't consider this as a drug molecule, and the drug response will be less reduced. And in case of preclinical studies, animal tests of toxicity and efficacy of therapy like will be considered in the rodents, mammals, and primates and mouse, and then it will consider for the clinical trial phase one on the humans and targets. Okay, and in phase one, volunteer healthy volunteers. Phase two, patients with dosage safety, and phase three, normal patients and to determine dosage and safety side effects. Both the perspective of the patient groups will be considered, and then it will be considered for the manufacturing and development of formula from this, and it will be formulated and it will be given to R&D for formulation, and then it will be based on the drug safety regulatory affairs, it will be marketed. And this is the overall drug development process. And later sessions we'll go for the docking studies. If you have any doubts, you can ask now.
Dear participant, if you have any question, please, you can unmute and ask your question. Dr. Grace, you can unmute yourself.
Thank you very much
Thank
for
you,
the
Dr.
lecture.
Grace.
I hope you can hear me.
Yes, ma'am. You are audible.
Yes.
Thank you, welcome.
So I have my question. I have so many questions, but let me see. When you
Yes, ma'am.
talked about the pharmacophoric features,
Yes,
I'm wondering some drugs may have hydrophilic features. So how do you factor that? I mean,
hydrophilic
when a
features.
drug, because you talk more on the hydrophobic features, what if a drug has more of the hydrophilic features? And you also mentioned that in the ADMET, this last one you just talked about in the absorption, distribution, metabolism, and the like. And I'm just wondering if a drug is more hydrophilic in terms of its features, how will
Yeah.
you how the application in terms of the pharmacophoric features you mentioned. That's number one.
Okay.
Then number two, you talked about the scoring. Now, if a drug is multi-targeted, how can the scoring be evaluated? It's not, I need to know. And
Okay.
finally,
Yes, yes.
you mentioned the ligand receptor binding driven by the hydrophobic interactions. Again, if a drug is more hydrophilic in its features, how can the ligand receptor binding be properly studied? And looking at the structure of some proteins not all proteins are hydrophobic. I mean, you look at the chains of amino acids, some may have, some may be philic in nature. So how can that be possible in terms of ligand receptor binding being driven by hydrophobic interactions? Thank you.
Okay, Dr. Grace, very nice to hear. Of course, good to hear the questions actually. First thing is like, first your question is like, you asked for the protein having or ligand having more hydrophilic in nature, right? In that
Correct.
case, yes. In that case, we have to discard it. It will not, we should not consider compounds with more hydrophilic in nature, because in case of absorption, if it is passing through a membrane, let us see here. If we are having a membrane, cell membrane, like you might be very familiar, ours is a bilipid layer, right? So I'm giving a rough diagram here. So so it's an bilipid layer. It's an outer membrane. Shall I get a new one? I'll show in a minute. I'll get a notebook here. Okay, I'll get in slide. No issue. Okay, let us consider this as Dr. Grace. I'm audible to you. Hello, Dr. Grace. Am I audible?
Yes, you are.
Yeah, yeah. Let's say this is our cell membrane. Okay, I'm just drawing it roughly. So bilipid layer. Okay. Okay, you might be the familiar with the membrane structure, right? This is outer membrane, and this is intramembranous space, intramembrane, and this is inner membrane. Okay. Here, if we consider a drug molecule, uh, some drug molecule, let us say this is a drug molecule. Your doubt is like if this drug molecule is more hydrophilic, right? How we can consider this molecule as a drug, right? If it is more hydrophilic, it will be dissolved in water present in the outer membrane. It will not cross the bilipid layer. It will not cross into the cell membrane, into the internal cell space. Cell membrane.
No, please, can I come in here? But that is why you have formulations, because this is actually another problem that we
Yes, yes.
see.
The problem is like you should consider the optimal hydrophilic nature is allowed, because within the cell membrane we have cell channels, ion channels will be there, right? Here we have ion channels, so those ion channels
Yes,
will allow based on the permeability and the concentration gradient, it will allow. Okay, for that allowing also it should have optimal range. But your doubt is like to have high hydrophilic. If it is more hydrophilic in nature, it will be get dissolved into the outer water environment. It will be dissolved. It will not get into the cell by crossing the bilipid layer, and it will not show its action. So it will be a failure drug. So drug molecule with high hydrophilic nature should be omitted. Okay.
yes.
Yeah. And then
I
I forgot your second doubt, ma'am. Can you repeat your second doubt?
Can you hear me?
Yeah, yeah.
I
Yes,
agree with you, but you see, we have this problem that in silico, you have the scoring, everything is okay. But when you come to physiological states, when you have in vivo, in fact, even in vitro, you
yes,
have that same prediction with a very high,
yeah, yeah.
promising prediction,
Okay, I
not
understood your
having good activity. And I
yes,
think it
yes.
has to do with all this
After
that
yes, yes.
in
I got
silico
your
studies did not actually
I
put in place. I'm not really saying
I got your
100%
doubt again.
hydrophilic,
I
but
got your
if
question,
a
ma'am.
drug has some hydrophilic features as well.
Yes, yes. I got your doubt right now. So if you're considering this as a good molecule with good docking score in silico methods, when we are going in virtual real method, real tech activity synthesis method, it is not showing its activity as expected in silico, right? As we are showing is a good one in silico method, is not actually representing the activity in virtual real time method, right?
Yeah.
Yeah. In that case, we have to modify it. We have to scaffold the compound from the compounds in silico methods. Very clear. What's the second question, ma'am?
The second question is having looking at the structure, just the same, the structure of protein. Some of them are not all hydrophobic in nature. So I'm still wondering.
Okay. In
Yeah.
receptor thing, if you're having more hydrophobic nature or hydrophilic nature, then what should we do? That's your question, ma'am.
Yes.
Yeah. In case of receptor hydrophobicity and hydrophilicity, if it is three dimension, the structure is predefined with crystallized structure. Sorry, if the structure is crystallized, we can get to know its properties. Like hydrophobic will generally will tend to find in the helix, and hydrophilic will tend to find the beta sheets. If it is more hydrophilic, it tend to find into beta barrels than beta sheets. In that case, these beta sheets and alpha helices will define the structure, right? Secondary structures will define the folding capacity of the receptor, and that foldings of the secondary structures ultimately define the three-dimensional structure and its function directly. So more hydrophobic in nature will not exist as a single structure, single protein. Either it will exist as a complex protein or it will exist with some other uh, ligand compounds or some activators or some stimulants will be there. So we can find some proteins that will be in more complex rather than in an individual protein. If it is more hydrophilic in nature or hydrophobic in nature, that will exist in as a complex structures in real time. So in that case, we have to consider the complex structure for docking studies, not individual structures.
Okay, thank you very much. But the last one
Next one.
is the multi-targeted drug. If a drug is multi-targeted, how will you be able to ascertain the scoring?
Okay. In that case, uh, we can consider things like docking of the same compound to the, for example, two targets are there. For example, protein A, protein B involved in diseases, some XY disease and some YZ disease. But the target A, target Z, I mean, like our compound drug molecule is having the binding affinity more to A. For the both the proteins, we have to compare the binding energy and binding affinity along with the docking score. Binding free energy should be considered, and most importantly, binding pattern should be compared between the receptor interactions. Multi-targets. If you're having two targets, target 1, target 2, interaction of the ligand to the target 1, interaction of the ligand to the target 2 should be compared in three cases, like binding docking interaction, well, like docking score, and binding free energy, most importantly, and more importantly, binding pattern between the targets. Based on this, we can propose single compound as a compound drug molecule for multiple targets.
Thank you very much.
Thank you. I think you have. I think I have cleared your doubts. I think then we can close today's session. Has been extended for more time.
Any more question, please? Jimmy George, right? If you have question, please feel free to ask.
Thank you so much. I think for now, everything is okay. As the session continues for the next 5 more days, these questions will be asked. Thank you so much.
Thank you, Jimmy.
Hello, good evening.
Good evening.
Very good evening.
Yes, thank you very much, sir, for this presentation. It's so wonderful.
Thank you.
I'm
Thank you for
Pradeep from Nigeria. Thank you
good.
very much.
Thank you
I
for
had
that.
my bench work in India, Pondicherry University.
Okay.
And so it's always a joy. It's always joyful to me to join my Indian colleagues once again. I have met them since I left last year, and now joining again now is a beautiful thing for me. So I'm really happy.
We too are happy.
Thank you very much.
We too are happy with you, and you can associate with us anytime hereafter. We are always
Yes, sir. Thank you very much, and thank you for these beautiful presentations. We are really learning great things. Thank you, sir.
Thank you for the compliment, and thank you for
Yes, sir.
look for the better for the next sessions.
Yes, sir. Thank you, sir.
Yes, thank you. And some Anaina Chinasa have raised some question. Raise your hand. You can unmute and you can ask your doubt.
Yeah,
Chinasa,
good evening, sir.
very good evening.
Please, I want to get more light. I would like to ask so you throw more light on the 2D, two dimension.
Okay,
How does
pardon me.
one practically go into doing that?
I'm not.
Like if I,
Sorry, I can't get your question clearly.
okay, I'm asking for the, my question is about the 2D, the 2D.
Like
Hello,
for 2D printing, ma'am?
I didn't get it, sir.
No, no. Actually, I'm not getting clear. 1 second.
Ma'am,
Asking
can you
about the
please
2D.
put your question in the chat box?
Okay,
That's
sir.
easy. Yeah,
Yeah,
because
it's fine.
not clear from my side.
Yes,
Not
yes.
your fault. It's from my side. It's what's not clear.
Any more question from the other participant? Dr. Srijita, Henry, Israel, Prince. Please, if you have any question.
Okay. Docking algorithms. Chinasa, you're asking for docking algorithms, right? I think you can unmute right now. That's what I have explained the types of algorithms in last 10
Yes,
slides.
sir. I'm asking for,
Yeah.
I'm asking about the talking algorithms, 3D.
Okay, 3D fingerprinting docking, right?
Yes,
Algorithm.
fingerprints, yes, sir. Yes, how do
Okay.
I practically, after I've gotten my target protein,
Okay.
probably
You just
how do I transcribe it now to
okay. You just imagine some beads are arranged in a. I'll just show you, right? Okay. Okay. Let's say we have one garland. These are with beads like this. One bead, second bead, third bead, fourth bead, fifth bead, sixth bead, like this, so on. Okay. This is, let us consider this one atom, second atom, third atom, fourth atom, fifth atom. Okay. If we can present here, atom is present, so we'll give 1. Here atom is not present, so we'll give 0. Here atom is present, so we'll give 1. Here absent, so 0, 1, 0, 1. So zeros and ones are nothing but bit strings. Okay, bit strings, bits, actually bit scores, bit values, 0 and 1. But if you're writing it like for a continuously, like multiple bit scores, 0, 1, 1, 0, this is a string. This is a bit string. Okay, this is zeros and ones are bits, and this is a bit strings. Bit strings. Okay. Bit strings. If this is can be done for 2D, like we are just simply we are writing bits, but in case of molecule, let us say is a benzene, simple structure benzene. Okay, this is simply represented, but while writing, we should write as This is also benzene, this is also benzene. But it's a 2D representation image. But while writing it three-dimensionally, how it exists, it will exist in three-dimensional like this. This atom position will be differ with the this carbon, right? First carbon to sixth carbons, alpha carbon positions will be differ, right? In X, Y, and Z axis. Okay. You imagine a three-dimensional, like in a space of here, yes, one double one is missing. Thank you for that. Okay, in a space of a room, or wherever you're sitting, you can just see the point as from the floor and the walls and the height. Some point, your eye vision will be straight. You will be present in three-dimensional space. You're not present in the 2D, right? Like this, these compounds, the position of the presence or absence of this atom here from some space will be defined. Okay, and thus will be given as presence of that atom when compared to the entire molecule in three-dimensional space. Present or absent will be given in the form of bit strings. So those bit strings were 3D fingerprints. Okay. See, let us see how our human fingerprints are unique. Right? Human fingerprints are unique. Similarly, each compound fingerprints will be unique. To compounds, atoms of a molecule distributed in three-dimensional space will be arranged differently based on rotational bonds, based on the torsion angles, based on static hindrances, based on the clashes or van der Waals force of attractions. Right? Based on all these, the 3D fingerprints will differ with the atom of molecule of atom to atom, molecule of different atoms, right? So that
Dr. Pradeep, your mic disconnected. Please check it.
yes, Wi-Fi disconnected. Okay. Yes. Here, like this, as a human as our human fingerprints are unique, the availability of distribution of the atoms or the molecule in 3D space will be unique, and that presence of I think actually you have network issue. So fine right now. Sir, is it am I audible right now?
Yes, you are audible, sir.
Okay. I think I've cleared that doubt, sir.
Dear participant, any more question? I think there is no more question. Thank you, dear professor, for your valuable thought, which you present in this session, and all the questions from the participant has been discussed. And it was really a very nice presentation, informative, knowledgeable to all the participants. We all enjoyed your lecture. Thank you very much for your valuable time as well. And thanks to all the participants for being the part of this session. Tomorrow, we will join the meeting. And if there is any change in the schedule, we will intimate you at least 2 hours before the session. Thank you very much. And good night from India to everyone. Thank you.
Thank you, sir. Good night, everyone.
Thank you very much.
Thank you.
Thank you.
Thank you so kindly.
Yeah.